Recent technological advances in biomolecular science and biotechnology are allowing us to synthesize DNA from scratch. These advancing methods are becoming cheaper and more efficient as time goes by, bringing with it a variety of practical applications, one of which is data storage on DNA. Join me on this journey while I will explain to you how data can be stored within DNA and why this technology has the potential to outcompete current storage methods. Now, before we delve into the biology, let me explain to you how current storage methods work, using the flash drive, aka the USB, as an example. Essentially, a flash drive is just a platform with millions of tiny transistors. These transistors are set up in circuits which interact to store information via a bit code. Let's have a closer look, shall we? So here we have one of many tiny transistors. These devices may vary how they relay the information, but their storage mechanisms are all the same. There is this oxide layer, sandwiched between a control gate and a floating gate. This layer has the ability to store electrons without the need for power. This layer can be charged, pushing the electrons through and relaying the bit information. These transistors can be organized to store very complex information using this mechanism. There are, however, drawbacks to using flash storage. For instance, if you were to damage and crack this oxide layer by, let's say, corrosion or by dropping it, this crack can lead to the leaking of electrons from the oxide layer, corrupting all of your precious information. The durability and the data storage capacity of these drives is enhanced every year, but the danger of data corruption by physical damage will always be there. Now, what if I told you that DNA can bypass this problem? Deoxyribonucleic acid, or DNA for short, is a biological molecule composed of nucleotides which code for all genetic information in all forms of life. It exists in a double helical form with four nucleotide compositions. First off, we have the purine molecule guanine. This purine binds with its pyrimidine complement cytosine. Secondly, we have the purine adenine, which binds to its complement tyrosine. So, you may be thinking, how can this biological molecule store our personal data? Sure, it can store genetic information, but how can we mess with it to make it store pictures, books, movies, and so on? Let's now dive into why scientists nominated DNA as a potential data storage molecule and the methods they developed which allow us to store our personal information on DNA. So, why DNA? Well, George Church and his colleagues described in their 2012 publication the advantages of using DNA as a new platform for data storage. Firstly, they reasoned that DNA had natural data storage capabilities. As we know it, it stores a complex array of genetic information. As a result, in nature, DNA is constantly being read and written through enzymes and other biomolecules. The second reasoning was its resilience. DNA can withstand a large range of temperatures without degradation. It's also been shown that the information on DNA can still be read after degradation. Lastly, DNA provides grounds for non-planar information storage. It can be condensed into tiny spaces, much smaller than the planar organization of a transistor circuit on flash drives. So with these different aspects in mind, let's have a look at how George Church and his team went about storing non-genetic data on DNA. They used the book How Synthetic Biology Will Reinvent Nature and Ourselves as a sample of text to store. The words within this book were converted into binary code or a bit code. This code was then further translated into a nucleotide sequence. Following this, the desired nucleotide sequences were synthesized from scratch, encrypting the data into nucleotide libraries. So what about accessing this information? You can do this by sequencing the nucleotide libraries using next-generation sequencing. The sequenced DNA can then be decoded, revealing the original bit language, and further decoded to reveal the original words from the book. So let's have a closer look at this process. So where would you start? To begin with, the words from your book can be rewritten into a bit language. Essentially, this language replaces all of the letters, spaces, numbers, uppercases, lowercases, and any other literary aspects of a typical book with this unique code. This bit language is just a combination of ones and zeros. You can further translate this bit language into a nucleotide sequence. George and his team did this by writing a code called bits2dna.pl, which allowed them to convert the whole book into bits, then further into nucleotides. They also incorporated a 19-bit barcode into each segment of their book to allow for rapid identification of each nucleotide sequence within the whole library. Overall, they encoded 1 bit per base, turning the whole 5.27 megabit book into 54,898 nucleotides. This sequence also avoided 4 or more nucleotide repeats and had a balanced GC content. So now you might be asking, how did they assemble all of this? So after they had their desired sequences determined, the DNA oligonucleotides were compiled using phosphoramidite chemistry, which is a 3-step chemical process which essentially adds single nucleotides on top of each other one by one. This process involves the deprotection stage, a base coupling stage, and an oxidation stage. Once the desired oligo sequence is achieved, it is then capped by acetylation. These oligonucleotide strings are then compiled on a microarray chip as oligo libraries with sequences corresponding to the bit code of the original book. Okay, so now we've discussed how you would go about storing information on DNA, but this storage is no good if you can't access it. Let's now talk about how George and his team were able to access their encoded information. They used next-generation Illumina sequencing, which is a very rapid way of sequencing DNA. First off, the DNA from the microarray library undergoes reduced cycle PCR amplification. Through this amplification, additional motifs are introduced to the ends of the oligonucleotides. The oligos are then isothermally amplified on a flow cell. These flow cells have oligos bound to their surface, which are complementary to the motifs which were added to each oligo during PCR. The oligos are then cluster amplified by different polymerases, resulting in clonal amplification of all nucleotide fragments. Following this, fluorescently tagged nucleotides are added to the single-stranded oligos, which are detected and allow for sequencing by synthesis. The emission length and intensity of each bound nucleotide is analyzed and determined using a computer software. The data of millions of different nucleotides is compiled and the sequences are determined with a high degree of accuracy. Following this, George and his team aligned the sequence data using the incorporated 19 nucleotide barcode and utilized their bits2DNA code to convert the sequence back into bit language and from there back into words. From this experiment, they were able to code 5.7 megabit bit stream into 54,898 nucleotides, achieving 5.5 petabits per millimeter cubed. This greatly exceeds current flash drive storage capacities. They also found an error rate of 10 bits per 5.27 million, proving that DNA can be a highly reliable mode of data storage. Even though DNA has a much higher storage capacity and a larger durability than conventional transistor circuitry, its data storage abilities present some pretty significant drawbacks. For instance, the actual oligosynthesis and sequencing are all limiting steps in this process. They are far slower than conventional methods. Furthermore, the cost of these technologies is very high, presenting yet another obstacle for DNA as data storage. With this being said, it would be sensible to use this method of DNA data storage for more archive-type storage of very large files. It is also important to note that DNA synthesis and sequencing technologies are advancing at a considerable rate and becoming more and more accessible as the years go by. In summary, DNA has the potential to provide a solid ground for a very large and stable data storage platform. Advances in our abilities to both synthesize and study DNA have shown us its potential regarding data storage. Unfortunately, the technologies which would allow us to perform such feats efficiently and in a cost-effective manner are still highly lacking. As I previously mentioned, both DNA synthesis and sequencing methods are becoming more and more accessible and cost-effective, allowing for DNA data storage to become a reality in the near future. Thank you for watching and I hope you enjoyed.